Hi, this video goes over the unique cell-based assays called pharmacotrafficking assays for membrane proteins like GPCRs and ion channels and other things like transporters. It allows you to identify small molecule compounds that rescue disease-relevant mutant membrane proteins. What we'll go over is three different sections. First, I'll start out with going over what cellular protein trafficking is and trafficking defects, and then how they relate to diseases with the focus on drug therapeutics. Then these are specifically called pharmacological chaperones, and how these pharmacological chaperones potentially correct these diseases. And then we'll go over the assay technology and follow that up with several different applications for drug discovery as well as subunit assembly type of applications. All right, let's get started. First of all, what is protein trafficking in the cell? Basically, it's the cellular movement of proteins going from different compartments, such as GPCR, for instance, being internalized into the cell to its endosome. You can think of this as being very essential to the biologic role of the cell, and it affects GPCRs, ion channels, even receptor tyrosine kinases, even other proteins such as NHR, or nuclear hormone receptors, and other proteins. If these movements are altered in any way, maybe by a ligand or therapeutic binding to its the receptor, or like a protein mutation, even like a deletion, or an aberrant type of signaling, undesirable effects may occur, and often resulting in drug tolerance, unwanted side effects, and even disease. So it's important to understand what is going on with your protein when the disease state occurs, for example. So what we're looking at here is a very simplified illustration or summarizing the progression of a transmembrane protein trafficking to its intended location, which is the cell membrane. The normal process by which proteins are folded and trafficked. So these proteins, for example, we're looking at a GPCR here, starts out obviously in the nucleus where it's transcribed and translated, and then it gets moved to the endoplasmic reticulum, or the ER, and then once it's properly folded, the proteins are translocated to the Golgi apparatus, where it undergoes post-translational modifications like glycosylation, and then it is trafficked to the surface of the cell membrane. So, however, sometimes defects do occur in the proteins, such as a mutation or truncation, as I mentioned, or deletion, and this can lead to a failure of proper folding, rendering the protein non-functional, and it doesn't make it all the way to its surface, for instance, to form its to do its function. How cells typically deal with this is via a quality control-like system which prevents misfolded proteins from trafficking properly. So instead of exiting the endoplasmic reticulum, they undergo two different things. It's part of the defective proteins process called ER-associated degradation. So it gets rerouted either to the proteasome, or sometimes it gets misfolded and it aggregates, which obviously is not desirable. So here's a list of protein misfolded diseases. It's a short list, but these are the ones that result from a genetic mutation or a single point type of mutation or a deletion that ultimately leads to the function or loss of function of that protein, possibly due to the improper trafficking in most cases, and ultimately giving a specific disease phenotype like cystic fibrosis, obesity, diabetes. I've got a few listed here. These particular diseases shown here in the first column are linked to a variety of different target classes, shown in the last column, like GPCRs and ion channels, as well as transporters. For example, let's look at the first one as a GPCR example that leads to hypogonadotrophic hypogonadism, which corresponds to male menopause or a loss of or low testosterone. This is linked to a specific mutation in this receptor here, gonadotropin-releasing hormone receptor. This topic associates with a large market base. For example, in pharmaceutical companies, they have spent more than $100 million over the last several years to promote treatments for this low testosterone. This is a statistic back in 2012, with gross sales in the U.S. specifically topping over $2.2 billion. So it is a huge market. So in this talk, we're going to focus on the applications associated with these specific targets or disease states that are starred all the way to the left side. Okay, so before we get into the technology and then the applications, it's important to understand what are these therapeutics. So to date, there's numerous compounds that have been characterized for their ability to correct protein misfolding defects or mutations and induce this forward trafficking of the protein to the cell membrane where they are needed to function properly. And these compounds are called pharmacochaperones. Many of the papers out there call them pharmacological chaperones. And they are obviously necessary to clear the disease state or rescue it. So let's look at kind of what they do. They can be defined as compounds that can penetrate through the cell membrane, so obviously they're small molecules, and directly bind the misfolded protein. And what they do is they promote the folding of that protein by stabilizing the membrane protein, compensating for its destabilization effects of that mutation. And then they facilitate the forward trafficking of that protein to the cell membrane, and ideally provide the ability for the function or the protein to perform its intended function and rescue the protein overall, leading to, most importantly, reversing the course of that specific disease. So actually curing it is actually kind of cool. So just wanted to give you some case studies, a quote and some clippings that was out during the first time, like when this approach became new in like the early 2010s. The approach, they highlight the quote, they highlight the discovery of this emerging pharmacotrafficking phenomenon and the realization of these pharmacochaperones that could significantly help with therapeutic value because of their potential in treating the actual disease by restoring its function rather than just merely treating the symptoms of the underlying disease. So let's focus in on this lower left-hand corner. The February 2016 article in particular talks about hereditary cataracts, and this is due to a single point mutation, R to 122 glycine, and abundant lens protein called the alpha-crystalline that stabilizes the protein, leading to its loss of function, formation of aggregates, obviously, and eventually cataracts. So they discuss in this paper the use and discovery of a pharmacological chaperone that binds and stabilizes this native state of alpha-crystalline and preventing the disease. So it's really cool. More recent example from Temple University this year, 2020, a study successfully showed a pharmacochaperone that helps with trafficking of proteins to prevent Alzheimer's disease in a mouse. So this is kind of exciting. All right, so before I get into the assays that come from Eurofins DiscoverX, there are several assays available that customers and researchers like you have been using as an effective approaches for monitoring pharmacotrafficking. There are certain limitations for each method, which I'll go over. So the first, or the most widely used, are these two on the left side. This is an imaging type of approach, or a FACS analysis, which stands for fluorescent activated cell sorting. This is, a specialized type of flow cytometry. They're both useful in detecting trafficking, but they have their issues with their low throughput, require good antibodies or epitopes or fluorescent tags for the detection, as well as specialized expensive equipment like flow cytometer. The next one, or the one on the right side, are functional assays, and these are great. They're useful for detecting restoration of the function and high throughput capability, but they can be very difficult to develop. There's not a lot available, And they're not always HTS friendly, even though it does have the capability for doing that. So there is clearly a need for an easy to use, high throughput type of compatible tool for studying these pharmacochaperones that doesn't require antibodies and all the other stuff I mentioned. So let's get into these assays. They're called PathHunter Pharmacotrafficking Assays. The basis behind these assays, like most of the products that come from Eurofins DiscoverX, is based on our enzyme fragment complementation technology. So if you look to the right side, you kind of see the basis of the technology, which is based on a beta-galactosidase enzyme. There's a really small fragment portion called the enzyme donor. In this specific assay, it's called ProLink. So you'll see like the protein being labeled with PK sometimes. And there's a larger portion called the enzyme acceptor. The two fragments have no enzymatic activity when they are separated, but together when they recombine or complement each other by bringing them together through a protein-protein interaction or other related type of event, they form an active enzyme, and that could be hydrolyzed in the presence of the substrate, and then allows you to emit the chemiluminescent signal, which can be read on any standard luminometer. So the pathway assays are engineered to express one or both of these specific fragments fused to a protein, which at least one of them is your target of interest, okay, your protein you're trying to target. And then the other is localized to an organelle like the membrane or the endosome or some cellular compartment, for instance, for other types of translocation type of assays. So these fragments can be interchanged, and it gives ultimate flexibility of the system to do other things, like protein interactions, translocations, and even modulation of protein levels. Listed some benefits of the technology here, looking at multiple applications for, small molecules as well as biologics. Not in this case because you need the pharmacochaperone to go into the cell, so it's focused on small molecules. These assays are definitely homogeneous, meaning they're not, doesn't require wash steps like typical ELISAs do, for instance. It's a mix and read type of format. They're fairly quick within a few hours. They're robust. They have large signal to background noise because this is an enzymatic amplified type of system, and they have high selectivity to their specific target. They're very sensitive and high precision, especially for our bioassays, which I will not be talking about today. They are definitely highly qualified and validated with our customers as well as internally in-house through our screening facilities. Got lots of publications around these, and then they're scalable from 96 to 3456 well-play format. So they're high throughput friendly. Here's just an example of the protocol where you plate the cells inside of your plate and then incubate overnight. You add your compound, incubate for a few hours, and this depends on the target and obviously the ligand that you're working with. And then you add your detection reagent, which has the substrate, incubate in the dark, and read the assay plate in a standard luminometer. And then finally do your analysis. Here's your dose response curve, looking at efficacy and potency, etc. of your compounds. All right, I won't go into these other details since they were talked about on the last slide. All right, so let's move into the specifics of this pharmacotrafficking assay. So in this case, we have engineered two different parental cell lines, okay? They're differentiated by their location of that large EA fragment. So here I'm showing you this enzyme acceptor that's bound to the membrane. This is the first format. So the EA is bound to the membrane, while our ProLink or enzyme donor is bound to our target of interest. So this is a mutated GPCR in this case. So what happens in this format, once you add your pharmacochaperone, penetrates through the cell membrane, binds to your specific membrane protein that has been mutated or unstabilized in some sort of way. Maybe it's misfolding. It stabilizes the system, the target, and which allows it to shuttle to the surface where the PK tag can now be in close proximity to the EA tag. So then the addition of the substrate, it has the enzyme complementation occurs and hydrolysis of the substrate, and then obviously a detecting of that chemiluminescence signal. So this is the first assay format related to the membrane, and the second assay format is here. It has the ProLink also associated with the GPCR, but the large enzyme acceptor is now bound to the endosome. So after the addition of the pharmacochaperone, the mutation binds to the mutated protein and subsequently goes all the way up to the membrane, as you've seen before, but then it subsequently gets internalized. And this is a natural recurring or recycling process for membrane proteins in general. And they recycle and then sometimes they come back to the membrane. So the reaction in this case is occurring at the endosome, so you can see, did it properly get internalized in this case? All right, so the endosome EA format right here is useful for targeting target membrane proteins that internalize when either activated or whether it's just, naturally biologically recycled. So next I'm going to show examples of screening pharmacological chaperones focused on GPCRs, and then I'll move into ion channels. So this is our first example of a GPCR. It's a mutation in the rhodopsin receptor called opsin. It's essential protein molecule for dim light vision. When it's mutated, unfortunately, it causes a disease called autosomal dominant retinitis pigmentosa. So this should be, it's a mouthful to say, but it's a common disease, but it's affected only maybe about 10%. So not as common as I mentioned, sorry. But it's due to an inherent instability of the mutation, a P to 23H, or to histidine. And it leads to enhanced degradation caused by this disease. So symptoms occurring like loss of night vision or peripheral vision. And it first appears in children. And then they have severe problems typically as they develop in life, so in adulthood. So in this specific paper, using this pharmacotrafficking assay, the researchers performed 8 different assays, okay? So they did not only the EFC assay, but they also did BRET, bioluminescence resonance energy transfer. They did high content imaging. They did immunoblotting, RLUC reporter type of assay, and activity assay, alpha ELISA, and dot plot. So they did a lot of different assays. So it's a nice comparison study, so you can compare all the different technologies. In the EFC specific pharmacotrafficking assay, they used this molecule or pharmacochaperone called 9-cis-retinol, which is an analog of the naturally occurring chromophore, 11-cis-retinol, to stabilize the mutant and allow it to do proper folding and trafficking to the cell membrane, allowing for the protection of the rod photoreceptors cells, and therefore were confirmed by also immunostaining to show that it went to the membrane as well. So they're showing it here, that it now lights up the cell membrane. So overall, the paper indicated that the assay was very simple. They said it also was easily adaptable for automation. It allowed for quantification of the translocation and highly reliable for HTS. So that was really nice paper. The reference is down at the bottom by Chenin and all. All right, this next slide shows 2 more examples or case studies of GPCRs. Both of these targets have multiple known natural mutations, okay? The results in the receptor defect, the trafficking, and leads to their specific disease. So let's start off to the left. We're looking at severe early onset morbid obesity in humans that has been linked to single point mutations in the melanocortin 4 receptor. So that's this MC4R, which is a regulator of energy homeostasis. In the market, it's become an attractive target or drug target, while mutations account for about 6 to 8% of the severe inherent obese symptoms, and they are relatively rare though in the general population. The symptoms include increased risk for weight gain and causing increased appetite and decreased satiety. In the right side, we also did a pharmacotrafficking assay we have available for this receptor. This is the vasopressin receptor, or AVPR2 receptor, and any mutation to this causes, or a specific mutation causes nephrogenic diabetes insipidus, which is kidney specific. This is a rare disorder characterized by the excessive thirst and frequent urination. It's unrelated to diabetes mellitus, just want to make a comment, and it's actually more common in pets. So here it's showing the assay using a specific drug, rilcovapatin, that is now in clinical trials. So next, I wanted to go over the ion channels, but if you don't see a specific pharmacotrafficking assay for your specific target mutation, you can always call us and contact our custom assay development team down here. There's a link at the bottom. All right, so now let's move into ion channels and transporters. We've got a few indicated here. It's a subset of all the ones that are known. The first example is right here. It's called cystic fibrosis, and it's specifically for a membrane CFTR receptor, an ion channel receptor, cystic fibrosis transmembrane conductance regulator. So it is a chloride ion channel with a mutation, specifically a deletion in phenylalanine, or F508. That's indicated down here. So deletion mutation. And for those of you who are unfamiliar with cystic fibrosis, it is an inherited chronic disease that affects the lungs and the digestive system. And basically it blocks the airways with a buildup of flattened cilia. It's kind of a nice picture down here with a thick and sticky mucus layer that makes it really difficult to breathe. So in the disease state, the cilia basically cannot be effectively sweep the normal mucus off the membrane, and it stays there. And this is due to lack of chloride ions escaping from the lung cells. So in the U.S., it still today afflicts about 30,000 children, mostly under the age of 2, and into it adults as well, potentially rising to about 70,000, I think, worldwide. The last time I looked, life expectancy for these patients are in their literally the mid to late 20s. So this is about 60 years ago. Many children with this cystic fibrosis died before reaching elementary school age. However, advanced treatments has helped these individuals live into their 30s, 40s, and beyond. So it is critical to create nice therapeutics to help the specific patients. Next is a case study that we worked on with Vertex Pharmaceuticals. Created a cell line with the membrane EA type of format and the PK tag to the N-terminus of a specific portion of the CFTR. So they're looking at the same one we just talked about. This left image is an immunofluorescent image showing the ion channel is mostly stuck in the ER. It's indicated right here. And then next, we wanted to introduce pharmacochaperones to rescue this mutant ion channel. There are a number of correctors or pharmacochaperones, they term it correctors in this example, that was used to develop this promotion of the trafficking of the CFTR receptor. But I'm only showing you a few here. Specifically 5 of them in the middle graph, where we profiled all of them, and you do see some level of rescuing or trafficking to the surface for all of them. And most importantly, the rank order was the same as indicated in the literature for all these specific correctors. So next, Vertex Pharmaceuticals, who developed compounds to rescue CFTR mutants, including this one called VX809 experimental drug that is being tested in clinical trials. So what they did is I have the same, and the black line is one of the correctors, C4, and then the blue curve shows the experimental drug effectively rescuing the CFTR in the assay. And then more importantly is the red one. The red curve shows the results of testing both of these in combination. So importance of combination type of therapeutics. So the data is interesting. It shows like an additive effect, which is actually the expected behavior of these two compounds. So overall, the case study shows the effectiveness of the assays in this pharmacochaperone activity. All right, next example is an ion channel involved in cardiac arrhythmias. And this specific receptor, which is a potassium channel, human ERG or ether agogo related gene, are associated with a condition called specifically congenital long QT syndrome, which leads to cardiac arrhythmias. Okay, so there's more than 100 of mutations in this specific ion channel. They have differential responses to sodium channel blockage, and mutations in the potassium channel lead to this increase in heart rate. So one specific mutation in the this ion channel is glycine 601 to serine, which causes an aberrant intracellular ion channel trafficking effect, leading to the cardiotoxicity. Reductions in this specific channel, or excuse me, the mutation causes, the potassium channel to be reduced, the cardiac repolarization, prolonged QT intervals, and lethal cardiac arrest. So it's very important to study this specific ion channel. And then we used compound chlorophyllum to lower or show effective of the trafficking and rescuing this specific ion channel successfully. And finally, our last example is kind of unique. It is an ion channel, but we're looking at subunit assembly of the specific ion channels, and you could do this for transporters as well. So we're looking at subunits. Normally, subunit assembly occurs in the ER, or always the proper, and then it's critical for it to assemble properly for it to traffic to the surface, the membrane. If any of the subunits are missing, there's little to none trafficking, and they remain in the ER, similar to the mutant receptors and ion channel experiments we previously discussed. So in this specific case study, we created a cell line with the voltage dependent, it was a calcium channel, CAV 2.2, and the membrane EA format in this case. And this particular ion channel has 3 different subunits called A1B, 2D1, and B3. They're also shown in the x-axis of this plot. So in this experiment, we tagged the ProLink or small enzyme donor to one of the subunits, specifically A1B. That's why you're seeing it in all these different the plot or bars. So looking at the bar graph, you see that one subunit assembly has some kind of a low basal activity, and if you add a second subunit, you still see this low basal activity and incomplete assembly going on. But adding the third subunit, all 3 of them, results in high basal activity, which is indicative of the ion channel reaching the membrane, where the ProLink can combine with the EA, creating the complementation, generating the signal that you're seeing here. Just noting down here, there's 2 publications around this application for your reference. All right, relevant areas though of interest for this specific application and monitoring proper assembly and trafficking include like natural mutations that result in misfolding subunits and preventing trafficking, or studies of like chemical disruptors of subunit assembly. Those are 2 common ones. All right, to summarize all the ones we talked about, this slide shows all the different receptors that we spoke about, and the first one in particular is just used as a control, so it's just a mutation in a regular GPCR. It doesn't cause any disease, so it's just use it as a control for the other ones. If there is a particular one though that you see missing or like one, definitely wanted to contact our custom assay development services. They can generate the cell pools, cell lines, and the full assay for you, and they do other services as indicated on here. So just visit the discoverx.com CAD page and to get more information and get you started. All right, let's end the video by summarizing what we've talked about. We talked about forward trafficking of the PathHunter Pharmacotrafficking Assays. It's a quantitative antibody and image-free type of analysis you're looking at. Chemiluminescent readout, we have cell lines, custom assays, and we even have toolbox products. So these are vectors that you can buy and create your own cell-based assay in-house for not only pharmacotrafficking, but a lot of other EFC-based assays. We talked about identifying pharmacochaperones that rescue disease-relevant mutant membrane proteins, correcting them or stabilizing them, allowing them to do proper trafficking to the cell membrane as well as into the endosome as well, allowing it for functional rescue. Applications we went over for different disease states for GPCRs and ion channels. So disease-relevant mutants as well as ion channel assembly type of applications. So with that, if you'd like to learn more, there's some links at the bottom. Feel free to visit or contact us through our tech support. And thank you very much.